Is this clear enough? Yeah? Okay. Let us start here, right in the middle. I even gave you little grid lines. That's nice. Okay, so meiosis provides the cells that's needed in order to perform sexual reproduction. So if an organism does not reproduce through sexual reproduction, then they have no need for this process. But most multicellular complex organisms do reproduce through sexual reproduction. So this is a pretty broad process that we see throughout the entire living world. What we see here is, in order to reproduce sexually, you would have two haploid gametes fusing in order to form a single diploid zygote. That single diploid zygote would then go on and divide and divide and divide, and eventually we'd have the whole multicellular organism. So what we're going to look at today is, how do we get these gametes? That's one. And two, what diversity is going to be present from one gamete to the next and why? We need to look at what's happening in S phase. There are some things that are occurring during interphase before meiosis really even occurs that helps to kind of get us towards this process. So first of all, a diploid organism is one that has two copies of each chromosome. Those copies are called the homologous chromosomes. We've talked about the difference between homologous chromosomes and sister chromatids before, but I just wanted to rehash that because we're going to be using both of these terms, and it can get really confusing if you don't have the right mental image. So during S phase, our DNA gets replicated, just like it does in mitosis. And because of that, we go from having one copy of every chromosome to having two copies of every chromosome. And since you have 46 chromosomes in your body, if you were to double that, then that means after S phase, we actually have 92 chromosomes. That means that's a cell. We call the same chromosome number from each parent homologous chromosomes. So like chromosome 1 from your mom and dad together would be considered a homologous pair. But the sister chromatids would be the two copies of one of those chromosomes. So I like this picture here. It's very simple, but it shows the idea very clearly. Let's make this up a little. The process of meiosis is going to give us genetically different haploid cells that we would call gametes. And this is going to allow us to permeate genetic diversity through a population. And you'll look at it over the weekend here or on Monday during Pirate Time, but that one video that I want you guys to watch and do that assignment on Canvas about genetic diversity. You'll see why it's so important to the survival of a population. If a population suffers a lack of genetic diversity, it is going to be highly susceptible to extinction events, highly susceptible to disease, loss of habitat, whatever it might be. Loss of genetic diversity is pretty catastrophic to a population. Another way that we know that genetic diversity is important to the survival of a species is like in the bacteria that we're trying to kill on a daily basis that are pathogenic to us, or even cancerous cells. So any cell that has a ton of genetic diversity in it, because their replication is so quick, that build up these mutations. The more genetically diverse a pathogen is, the harder it is to kill it with our current version of antibiotics, because they might happen to contain genes that provide traits that prevent them from being susceptible to that antibiotic. Same deal with cancerous cells. Cancerous cells, as they divide, they accrue these mutations. The more genetically diverse that tumor is, that profile is, the harder it is to treat those cancerous tumors, because they might not respond the same to each different treatment. So genetic diversity really provides a bunch of resistance to mass death through a population, whether that be good or bad for us. It just is. All right, 10 seconds here. Awesome. Okay, there are 3 main ways that meiosis provides genetic diversity within a population, so within the sexual reproduction pathway. And those 3 ways are homologous recombination and crossing over, independent assortment, and random fertilization. So we will start with homologous recombination. This is the idea of crossing over. Crossing over is specifically where the chromosomes like overlap. Okay, like that's the process of the overlap. But if you want to talk about the actual exchange of the genetic material, that's called homologous recombination. This occurs during prophase. And what we see during prophase is that those 4 sister chromatids of a homologous pair, so like mom's 2 copies of chromosome 1 and dad's 2 copies of chromosome 1, they will come together and they'll form a tetrad. We call that process a synapse, where the homologous chromosomes all come together. And then on certain regions, we will see overlap between the 2 middle chromosomes in that tetrad. Now, where they overlap is random. They can overlap in one place, which would be called a single crossover event. They could overlap in 4 places, which would be, an extremely diverse crossover event that we rarely see, but it is possible. Double crossovers are pretty common, though. And where they're overlapping, we will see exchange of that DNA. Now, there is a name for the place where they overlap. The location where crossing over occurs is called the chiasmata. Which kind of sounds like something you'd say when you perform a martial art move. So again, this is all prophase. Homologous chromosomes come together, form a synapse. That structure is a tetrad. They will overlap or perform crossing over in random regions down the 2 middle sister chromosomes, or down the 2 middle homologous chromosomes in that tetrad. We'll exchange that DNA. That exchange is called homologous recombination, because we are recombining the DNA on the homologous chromosomes. And that provides genetic diversity right there, because now we just took a paternal and maternal chromosome and we stuck them together, we blended them. You can see that this immediately would provide 4 different versions of the same chromosome number. So now I have 4 different versions of chromosome 1 that can be randomly sorted into a sperm-rearing cell. Can I slide down? So here's what this provides us. Imagine we just have a single crossover event. So now our end result is going to be 4 homologous chromosomes. One would be all of mom's chromosome 1. One would be mostly mom's chromosome 1 with a portion of dad's chromosome 1. Another one would be mostly dad's chromosome 1 with a portion of mom's chromosome 1. And then our 4th option would be all of dad's chromosome 1. So now we have 4 different versions of the same chromosome that code for the same genes. They're just going to have different genetic information for each of those genes. Now, multiply this occurring over all 23 chromosomes, and you can see a huge amount of diversity just homologous recombination and crossing over provides. But meiosis is not done. Just wait, there's more. Alright, who needs 10 seconds here? No problem. This process that provides genetic diversity is called independent assortment. You might need to paraphrase and squeeze here. My box was a little bit taller than yours whenever we printed. So independent assortment takes place during metaphase and anaphase. And here's the idea. When those tetrads align down the center of the cell at the metaphase plate, the order that they assort in is completely random. And what I mean by that is, you can imagine these things are stuck together as tetrads. Whether mom's or dad's chromosomes end up facing one direction, facing one side of the cell or the other, where they're about to be pulled, is completely random. And we'll sketch a little picture of this to kind of show you what I mean. But because of this, not only do we have 4 different versions of each chromosome number, but now we are going to randomly combine these 23 chromosomes together. So you might end up with a chromosome 1 that's mostly dads with a little bit of moms, and then chromosome 2 that's all moms, and then a chromosome 3 that's mostly moms with a little bit of dads, and then a chromosome 4 that's all dads. Like, it's going to be completely random how these things combine whenever the gamete's actually formed. Again, that is independent assortment. Each chromosome is going to assort randomly to either side of the cell. So the odds that every single chromosome that ends up in a sperm cell or an egg cell came from only one parent, and that combination didn't have any crossover events in it, so it was all the fully intact chromosomes, is absolutely astronomical. And this is why you don't look like, this is why you don't look like an exact clone of half of one of your parents. You might have a couple traits that are really, really similar, right? Because you may have inherited like all of mom's chromosome 2, for instance. But overall, you're not going to have a full set of one of your parents' chromosomes. It's going to be a blending of both parents.
Is this why like incest breeds a lot of effects?
Oh, yeah. So the problem with inbreeding is that any kind of genetic mutation that is present, there's not going to be any second copy to override that. You'd end up inheriting 2 copies of that same mutation. And over time, that can drive that mutation to become super prevalent within a population.
When we visited the sanctuary,
Mm-hmm.
all the life sizers, stuff they had Down syndrome.
Yeah.
And their eyes had like defects because over time to keep the bred cured.
Mm-hmm.
Like, it's just,
Yeah, and that's why so many dog breeds have just ridiculous amounts of genetic issues. Yeah. Even extreme overbreeding is a problem. Can I slide this down to here? Is that too far? Is that good? So if we were to sketch this out, you could imagine that we have, your tetrads forming, and I just did 2 chromosomes here instead of 23. There's not room to do 23, but the way that these things align is random. You could have dad's chromosomes on one side or mom's chromosomes on one side for each individual chromosome. So then whenever these separate, they're going to separate and sort randomly, and at the end, you would have completely different gametes that are just as likely as others. So certain combinations, or all these combinations, are just as likely as others. There's an equal probability, because there's just a 50-50 chance that they line up on one side or the other. So now we have 4 different combinations of every single chromosome, 4 different possibilities for every single chromosome, and we have randomly assorted all of the 23 chromosomes into a gamete. So they're sorted randomly, and they are all genetically different, each copy of each chromosome. So crossing over and independent assortment. Between these 2 processes, we end up with completely different gametes. So one round of meiosis provides 4 completely different haploid gametes. But there is still one more trick that sexual reproduction has up its sleeve to ensure genetic diversity in the subsequent generation, and that is through the process of random fertilization. Let's scoot this down a little. So both of these are just as likely. Okay, 10 seconds here. We're so close. I don't know if we're going to get there though. It's going to be tight. Good. Next slide. All right, last process. Random fertilization. So what we see is that during fertilization, no gametes are more likely to fertilize each other than any other gamete. It's a completely random combination of these gametes. So not only are each of the gametes genetically different, but the one that actually performs fertilization is randomly selected. So the combination of all 3 of these processes is what drives these huge swings in genetic diversity from one generation to the next. Even though you're starting with the same genetic material. It's just the way that we're wrapping it together, combining it, organizing it, and then delivering that material. Now, when you sketch this, I'm not saying that these are eggs and sperm from the same individual. Okay, so this egg would have, received its genetic material from their parents, and then the sperm would have received their genetic material from their parents, and they are going to be randomly combined. Can I scoot down a little? Can I scoot down to there? Is that too far? So any of those sperm are just as likely to fertilize the egg as others. Now, with this whole idea of in vitro fertilization, one thing that people are doing is they're starting to be selective as to which sperm and eggs they're combining. So what they'll do is they will test the genetics of the gametes before combination, or before combining them, and that way they can be more selective as to what types of traits they'll see in offspring. And this is something that a lot of people have an issue with if we do it with humans. We do it all the time, though, with like dog breeding and stuff. And then we just have a final statement linking this to the idea of evolution at the end. It's in blue down there. Can I shift down a little bit? Are you onto the diagram at least? I think I can get everything in view at once now. So that final statement says, genetic variation throughout a population is a major driver of the evolution and adaptive capabilities of the population. By introducing new gene combinations, which can aid in the survival of individuals within that population. So let's say that there's some big event that occurs within an ecosystem. I don't know, like there's a major drought or something. There could be some individuals, due to genetic diversity, that are better suited to surviving in a drought than others. And that's going to allow them to be able to live long enough to reproduce and pass on those genes, or potentially pass on those genes that allowed them to survive. So that genetic diversity basically would have made that population resistant to extinction from that drought. And that's where genetic diversity really has some power in helping the overall health and fitness of an entire population. Can I scooch? There? Awesome.